But if they can get a kick, they can go uphill. And that's what happens in chlorophyll. They get the kick from light energy. And this may happen in connective tissue. That is to say, the connective tissue light gets through the skin. And so photons are actually right now impinging upon our connective tissue, upon our collagen, and exciting electrons from one energy level to another. I want to say that again because this is basic to. I think this is basic to Ralphing. Let's look at the band structure. This is the band structure. Will just represent the different electrons in collagen. Collagen consists of carbon atoms, oxygen atoms, hydrogen atoms, mainly, and each of these atoms has electrons at different energy levels, so it's a system. And these are the energy bands. There will be electrons here, here, and here. These being the more energetic electrons. Now, there will also be bands, energy bands, which are empty. And there are many different energy bands that are unoccupied. And what happens when light strikes an electron, when light imparts its energy to an electron, is that it can jump to a higher energy band. And in fact, it can jump to a band where there are no electrons. It can jump to an empty energy band. And these bands are called, these empty bands, of course, after the electron jumps into that band, it's no longer empty. It contains one electron. An electron that jumps into an energy band, empty energy band, becomes a mobile electron. This is also called the conduction band, which means that an electron that arrives in that band can move very rapidly. So suppose a photon imparts its energy to an electron in this band. If that energy is sufficient, exactly enough energy, one quantum of energy, the electron will jump up to the conduction band, and then it can move very rapidly. It can also jump back down. When the electron jumps back down, it gives off energy, and this can be in the form of a photon coming off. I suspect that this is the origin of the aura. That is, this is where the light photons are in part. This is in part where these photons come from electrons that are jumping around between different energy levels. Yeah.
This seems very interesting, and Rolfers are certainly interested in enlightening their clients, but after all, we put mechanical energy in, not light energy. How does that connect?
Okay.
I mean, if I was shining a
okay, I'll tell you what happened. That question comes at the perfect time. Thank you. Let's go back to this model. What happens? I'm gonna put the microphone down and come over here and shout. What happens when we stretch this collagen molecule?
Diameter decreases.
Its diameter decreases?
Yeah.
Good. Thank you. That's what happens, right? Exactly. It gets longer and skinnier. And also there's a rotation. As that happens, the angle of the helix changes. Now this is too stiff to actually do that. Although when I look at it closely, I can see a slight twist occurring when I push on it. Now, what we refer to as tension in the organism, the resting tension, the stress level of an organism, represents a certain resting or a certain tension on this helical system, which will represent a certain angle to this helix, a certain degree of twisting. Okay, that will be the resting situation. Anytime we stretch it, we will change that. So in the process of stressing a collagen molecule, we will change many things. We will change the relationship between the individual strands. We will change the angle of the helix. We will change the angles between the bonds, the bond angles between all of the atoms within that molecule. They will be altered. The bond distances will change. And this will affect the energy band structure, using the solid state physics terminology. This will affect this band structure. Now, what I'd like to do,
when you stretch it, are the adjacent molecules sliding by each other, or actually what I mean to say, are the individual amino acid groups that make up that strand, are they sliding by each other so that like there would be an opportunity for an electron on one of those amino acid groups to get near another territory and be
exactly, yes, exactly. What is moving? Okay.
Right.
What does not seem to change, and this has been worked out very precisely using X-ray diffraction. You recall that these individual tropocollagen molecules are stacked in what I call a quarter stagger array, and they're cross-linked this way. And these cross-links are pretty strong. And it does not appear that they break during a really strong stretch of the collagen. It's unless you really put a lot of energy into it. They do not appear to break. It's not the main change in the system when you stretch it. Does not seem to be the breaking of these bonds. It seems to be the elongation of the molecule, just as I showed you, which causes a change in the helix. It gets longer and fatter.
As it does that, it like acts like a pump, an electron pump, to shift one electron here and then go back and shift it again and back.
Well, yes, as this occurs, it rearranges the order within the molecule. So now we're getting down to order, and what is order at the molecular level. These are two X-ray diffraction patterns. Now I should tell you a little bit about what X-ray diffraction is. Maybe I should figure out if I've talked too much. Am I talking too long? I mean, what's happening here? I have no idea what's happening. Okay, X-ray diffraction is a way of finding out the distribution of electrons within a system with a high degree of order. You can't do X-ray diffraction on a pile of stuff that is amorphous, that's not, in jelly. But the connective tissue system is a very highly ordered system. It's just, my mind is boggled at the degree of order that we have. We do not usually feel, when we feel our tissue, that it's a crystal inside of there. But in fact, it's very much like a crystal. The individual molecules are very precisely arrayed, and all of them all form a very elaborate, highly structured system. And this means that you can shine a beam of electrons through collagen, through connective tissue. You need a lot of molecules to do it. You can shine a beam of X-rays through connective tissue, and you will get a pattern, an image of dead spots and light spots. And these occur because the X-rays interact with the electrons in the atoms of the connective tissue. And from this rather obscure-looking system of dots and blurs, the X-ray crystallographer can go back and reconstruct the structure, the original structure, at the atomic level of the molecule. And on the left is unstretched collagen, and on the right is stretched collagen. And I think this was published in, 1953 by Cowan, and this has been repeated many times. What has happened is that the order has changed. And an X-ray crystallographer would say, this stretched collagen has a lot more order to it. That is, there's a lot more orientation to it. The atoms have lined up, have organized themselves in a precise way, which was more precise than in the unstretched collagen.
What kind of section we got there?
What kind of section?
Yeah, is that cross-section?
This is an X-ray diffraction pattern, which is the dark regions occur because X-ray photons, and X-rays are like light, they come in photons. X-ray photons have gotten through a piece of connective tissue, and they've made this spot dark on the photographic film, and no X-ray photons have hit the light areas.
Stretched is what I'm trying to find out.
Pardon?
In which direction has it been stretched?
Oh, this has been stretched in the longitudinal direction.
Which is up and down, north-south.
Right. Yeah.
It's my experience that there's that in a lot of ways the quality of change in the tissue from applying physical pressure by the process that we call rolfing is very different from the kind of stretching that you get from yoga. And I'm wondering if you have any ideas about that difference in that kind of stretching.
The question is it the difference in the kind of stretching you get with yoga versus connective tissue, rolfing or other types of techniques that deal with the elasticity and the stretching.
That the manipulation of connective tissue with our hands is in a lot of ways it seems to me to be different than if you just were to mechanically stretch that tissue.
Right. Well, I can't say anything at this point about precisely what rolfing does. I mean, it would be very nice to have an X-ray diffraction pattern before and after rolfing. That would be wonderful. Because that would give us precise molecular information or atomic information on what we have done to the order.
Is that even possible to do?
I don't know. I'd like to. There must be some way of doing it. Well, I'm sure it's possible to do it. I would prefer to do it in a non-destructive way. I don't know if you would have any success getting volunteers for that experiment. Yes.
Can I ask you a question?
Sure.
I see from the first picture that there is, not that much activity, or is there more activity?
Activity.
Yeah.
Okay.
What amino acids and what's inside the molecules? Because as you can see when it's stretched, you either have more open areas or closed areas. I'm not really that good at that to be able to tell the difference.
Well, I'm sure your impression of this, your subjective, maybe artistic impression of what the difference between these patterns is probably relevant also to what's happening at the molecular level.
You know what it is?
Okay, activity. I think what I should do is read a sentence from the Hunt report because I think it's relevant to this.
I think she's asking if the black is holes or whether it's holes or stuff.
I think you should look upon this as a difference in order, and you should realize that the chemist has a technique for measuring the order in a complex system, such as connective tissue. I should also tell you that this change in order, this increase in order, represents information. This is a change in information content. And in a sense, this may be the basis of, this could possibly be the basis by which connective tissue remembers what has happened to it. In other words, there's a change in the order of the atoms which occurs when the connective tissue is stretched, especially if it's stretched violently.
Is there any data in terms of what the distribution of stretched versus unstretched collagen is out of a living organism? I mean, do you find generally in one form or the other?
Okay, the question is whether there's been any work done on the living organism, right?
Yeah, any you know.
One of the difficulties is that most of the work that's done on collagen is done on isolated, purified collagen that's been extracted from the tail of a rat or the hide of a cow or someplace that's a good source of lots of collagen. So one has to be very careful in extrapolating from this work, which is done on pure collagen, to the situation in the body where not only is the collagen perhaps different, but it's also associated with other molecules, such as the matrix material, which is the subject of another lecture.
So we don't really know what form of collagen exists in a living organism.
We don't know anything about exactly what's happening in an organism because of the uncertainty principle. Anytime we try to measure something, we have to do something to it which changes it. So the best we can do is the best we can do. We do the best we can. Yeah.
Yeah, are these X-ray diffraction pictures of single collagen molecules?
Looking at a system, say 1 million collagen molecules, is equivalent to looking at one.
Because they do generally line up.
Right.
So according to that, what you have over there, the quarter,
Right, a quarter stagger array.
each molecule lines up in a quarter stagger array, generally.
Right.
Now, is there any distortion from that usually at all
Is there any distortion?
from the quarter stagger array? In other words, could this X-ray diffraction indicate that stretching causes a more linear quarter stagger array?
Well, I think the quarter stagger array does not change in stretching. I think the change is in, as we discussed, is in the helicity, the degree, the angle of the helix.
Now, in this quarter stagger array, those individual arrows, they're each a collagen molecule?
Each of these is one tropocollagen molecule. Right.
Okay, and so there may be
Millions of millions.
lined up like millions more arrows.
Right.
And they almost are never distorted.
Well, now, I don't know. I'm sorry, I can't tell you precisely what disorganized tissue is. Wish I could. I don't know what happens when two collagen molecules, when they become in a disorganized system. I don't know. Well, I know with a rope you can tie knots in it.
Yeah.
And I've seen electron micrographs that show little knots in the collagen fibrils.
Yeah, that's
And maybe Rolfing unties some of those knots. I really don't know. I have no idea.
okay. I have one other question, and that is does stretching affect the bands that we talked about earlier?
Okay, that's the next thing I wanted to tell you.
Oh, okay. I thought you were
Yeah, I was trying to, Gio.
Yeah. Before going on, when I was talking to Jim yesterday, I realized every one of us has seen what he's talking about every time we do a session with clients. Because if you see spirals around clients, basically we're getting more length along the longitudinal axis of the body. The angle of those spirals is getting more and more towards the vertical as you start getting a person lighter and taller. So what you're seeing out there, which is evidence of the shift in the angle of the helix at the molecular level, is really what you're doing all the time on the gross and macro level with the whole structure of the human being, the Rolfing process. Very nice little as above, so below analogy. And in terms of the geometry of it, it's exactly the same kind of process. Maybe that'll clear it up for some people.
I'd like to make an announcement. It's getting late, and I have a feeling that we're not going to be too good after this and the heat in here. So I've talked to Rachel and to Jim, and what I'd like to do is reschedule Rachel, who has to leave early tomorrow morning, for 8:30 in the morning, for those of you who are interested in body image. And then Jim will take the 10 o'clock spot that Rachel had. Is that clear to everybody? Yes?
Yes.
Thank you. So after this, we're going to be through for the evening until the party at 8 o'clock. Thank you.
How long do you want to go down? I'll keep us watching.
Whatever. Huh?
How much longer are you planning to talk to do?
Agree on a time.
I'm delighted to go on as long as you can stay, but I'd sort of like to finish it off in about 5 minutes if I can, because I want to put all this together and try to tell you what I think is happening with band structure. Yeah.
You made a comment about the change in the order of the atoms and the information there, and that it had to do with the degree of violence that was involved in the change in structure. Will you say more about that?
The question concerns the relationship between the degree of violence that's by which the tissue is manipulated, and I have nothing to say. I'm sorry. I don't know anything about it. But I'm sure that the work has been done, and I've not completed my survey of the literature on collagen by any means, and I may come across that, or maybe somebody else knows some biophysical data on that question of violent stretching. Yeah.
Are you through with the slides, Jim?
Yeah.
Yeah, I've seen some evidence that connective tissue is piezoelectric, that putting mechanical energy in transforms electrical energy. And it seems to me that may be the source of some of the electrical energy that happens with rupture. Do you have anything about that?
Okay. Well, I don't know. I think I should just finish what I want to say, because I think some of your questions will, you'll have a lot more questions after what I want to say relates to most of these questions, which you seem to be coming up. So why don't I just say it? Okay? I want to read from the abstract of the Hunt report. In the early Rolfing sessions, this is talking about the arc field. In the early Rolfing sessions, the field became random, dark in color, and low in frequencies. In the later sessions, even, coherent, translucent, and in higher frequencies. Okay, I want to refer to this business of the change in color of the field, which seems to have been established by this study. By low frequency field, this would indicate energy bands that are close together, so that when an electron becomes excited by whatever means, and there are several different ways that excited electrons could be produced in collagen, by light coming from the outside, or by light coming from nerves. And I should tell you that recent work is actually showing, it's actually been possible to detect photons coming from nerves. And I don't know if people have studied bones or connective tissue, but it very well may be that light is emitted by these tissues as well. And I can give you references to this work in the literature.
I believe in our eye is beginning to be measured. Is that the gleam in our eyes or
The gleam in your eyes?
Jim, just to wrap that one up, collagen has been found to be photoelectric, that is, when you put light in, you do get electricity out, and all those solid-state mechanisms theoretically are reversible if the conditions are right, and whether those conditions obtain in an organism or not hasn't been established, but there should be a way of driving that the other direction.
So that the flow of electrons will result in light coming
Flow
out.
photons back out,
Yeah.
that's right.
Okay. Now, this is sort of bringing the R into the physical, into solid state physics. Now, in the early sessions, then, according to this work, the light is low frequency, which would mean towards the red, green, yellow, orange, towards that end of the spectrum. And apparently these observations were made by Dr. Verrier. Okay, now the shift to a higher energy after Rolfing to the blue end of the spectrum would indicate a wider separation between the energy bands. So I've drawn this one higher than this one. I'm sorry, I didn't finish the story. The electron jumps up to this higher energy band, and then in the return back to the lower energy is when the light photon is emitted. So, if the transitions after Rolfing become towards the blue end of if they go towards the blue end of the spectrum, we start getting blue photons coming off. This indicates a change in the band structure. In the electronic band structure, a separation of the energy levels further apart. The way this would be brought about is very simple. I mean, the change in the helicity occurring on mechanically altering the length of the collagen molecule and possibly altering, well, I don't know, I guess I get the impression from what Gio just said was that collagen molecules may be lengthened during the Rolfing process. And that would, any change in the length, whether it's lengthening or shortening, should alter the band structure because it changes the relationship between the individual atoms. It changes the bond lengths and the bond angles and also the band structure. Now, to carry this one step further, the Rolf body, and just looking at this, now this is an interpretation, and I'm leaving the data behind, I think, in making this interpretation. So what I'm doing is offering you something to think about. That the system, we change the band structure of the system. But this is what Rolfing does. This means that more energetic electrons will be moving about through the system. This may be what we mean when we say that people have more energy, that they have a better flow of energy, that it flows from one part of the body to the other in a better way, and things work better. So to carry this one step further, it would say that the system is functioning at a higher energy level because of the greater separation between these bands. Yeah.
I guess I don't remember the report exactly, but it seems that in the end, what you're seeing was more of a cream white aura than blue. Is that not true?
A cream white, not a pure white.
Yeah, but white light, of course, is
Peach
all
color.
rather.
Peachy cream.
They were pastel tones instead of pure tones.
Yeah, but I'm just thinking in terms of the shift in band structure, you would definitely want to see a blue rather than a cream white, it seems like.
Not necessarily. I don't know what... I mean, white light, of course, is... Well, there's more to the story than this, of course, because what is happening also is a change in the electromagnetic field, which will interact with the light energy field. And I don't know how these, I don't know exactly what would happen there. Well, anyway, this is what I want to. This is what I want to. Where I want to stop, I think, and get your feedback on what I've said, and then tomorrow we can talk some more about it. Yeah.
I'm just curious, is St. Georgie mostly concerned about the flow of electrons through the length of a molecule, or from one molecule to the next, or both? You know.
Well, maybe I could I show another slide?
Sure. On the next one? Thanks, Jim.
Okay, this on the left is the latest model of collagen that I could find. That was published by an Indian named JimOshmanSatAnnualMeeting from Madras. And it's not too different from the other one I showed you before, except it has, he disagrees slightly on the arrangement of the hydrogen bonds. But this is a tropocollagen molecule, and this is the part of the circuit for my stereo FM AM receiver. And a consequence of the discovery that proteins are semiconductors is a relationship between these two. Semiconductors are the components of your pocket calculator and your tiny television set and so on. Semiconductors are conductors which can be very precisely regulated. This is how all of these solid-state devices work. This is how your pocket calculator works. There are lots of local circuits which can be, the current flow through these circuits can be altered very precisely, very rapidly. What's emerging from Dr. St. George's work is that living matter has similar properties to an electronic circuit. That is, it has diodes, which are elements that allow current to go in one direction only, which rectify. There are probably local oscillators, which allow fields to reverse and produce electromagnetic signals. There are capacitors, there are transistors, which they're probably organic molecules, organic components to our flesh, which correspond to the elements to a very sophisticated circuit. A kind of circuit that can not only do lots of things, but that can store information. And this is very exciting. This is, to me, one of the most exciting aspects of St. George's work. I don't know if that answered the question that was asked.
Well, is that, I just want to know if that's within the structure of that tropocollagen molecule or between molecules.
Okay, likely the elements of this circuit, and someday we may have a circuit. We may be able to draw out a circuit for a bit of collagen. And in fact, we're not too far away from that, thanks to St. George's work. The cross-links between adjacent peptide or protein strands would represent elements of the circuit. Each of the cross-links would be one element of the circuit. And there are different kinds of cross-links, so there are different kinds of components. Also, there are cross-links between adjacent tropocollagen molecules. As the structure is built up of many molecules, there are cross-links between adjacent fibrils and between adjacent fibers and so on. So all of these would be elements of the circuit. The control of collagen, the control of the flow of current of electrons through the system, Dr. St. Georgie has isolated the control points. The control points are the amino acids lysine and arginine. And they are the points where ascorbic acid, vitamin C, and other closely related molecules are able to hook into the protein and pull out electrons. And what happens when electrons are pulled out of the protein is that it then becomes a conductor. In other words, the protein, in terms of electrons, a protein, an analogy would be a line of cars that are waiting to get past an obstacle, and there's the traffic jam. And none of the cars can move. That's the situation in a protein where the electrons correspond to those cars. If you pull out one car, if you just pull one car out of the line, all the others can move. And that's the basis for the electronic conduction through proteins such as collagen. The normal structure would allow no movement of electrons, and as soon as you come along with an ascorbic acid molecule, you can pull out an electron, and then everything can shift. And this conduction, in terms of solid-state physics, is called Hall conduction. That is, it's the electron, it's not the electron that moves, it's the space that the electron occupied. The hole moves. And then all the electrons shift as a consequence of that. Yeah.
With the molecule lengthened as a result of having the electron removed.
Would the molecule lengthen as the result of the electron moving?
No, being removed.
Or being removed?
See, I don't think it would, but I think maybe it might.
Well, Dr. St. Georgi thinks that the stiffness, the stiffness of a protein would be affected by its being able to conduct electrons. And he thinks that proteins that are able to conduct electrons are more rigid.
That are able to conduct electrons
Right.
are more rigid than proteins that are unable to conduct electrons.
Right.
What did you say about the space? First you said the electron is actually removed as the car being removed, and then you said what actually happens is the space.
Well, there are two ways you can look at it. I mean, it's rather it's kind of trivial in a way, but the electrons can move. If the electrons move this way, it's equivalent to the spaces moving that way. And I think you need to get a bunch of billiard balls and play with them or something like that to see that. And if you, yeah.
Is tropocollagen generally pretty conductive then, or pretty? Is it easy for vitamin C to pull out a electron out of that?
Well, it appears. Now, I'm glad you asked that, because another of the findings of the Hunt study was that there's a change in the electrical the voltage measured between different points on the body after Rolfing. They put electrodes on different parts of the body. In one case, forehead and back of the head, and they found a higher voltage after Rolfing. So this indicates a change in the electronic structure, which isn't too surprising. I mean that would be consistent with the other things that we've just discussed. Also, in that study, high frequency signals were picked up, which corresponded to particular colors of the aura. And these high frequency signals probably arise because of oscillations in the flow of electrons through the collagen system, through the connective tissue system.
At the system level, rather than a particular molecule, it's like flowing through the whole body rather than small, sort of oscillating signals.
Right. Of course, this is probably due to a change at the molecular level. So these oscillations then become a matter of considerable interest, and as a biologist, I would certainly like to know the origin of these oscillations. And it probably has to do with some of this microcircuitry that exists in in the connective tissue structure. Yeah.
Tomorrow, maybe, could you talk a little bit about the relationship of light and electromagnetic fields?
The relationship of light and electromagnetic fields. I don't know much about that. I know a little bit about it. I know... Pardon?
That's about all I could absorb, I guess.
Oh, okay. Well, maybe what I have to offer will be harmonious with what you can absorb. Because it'll be funny. Yes.
I have two questions. One, is there any... What happens with heat in all this? Where does heat fit in?
What happens with heat?
Is there any heat or temperature change?
That's an excellent question. One of the ways of putting energy into a system is to heat it up.
Yeah.
When you do that, everything moves faster. It's, I guess some of that happened last night. At that hotel with Eldorado.
For sure.
What heat is a form of light. That is, heat comes in photons. I'm glad I can say that. I've been wondering about that for a long time. Heat is in the red end of the spectrum. Infrared lights, are hot. They're warm. Well, what they're doing is, those are warm photons. It's towards the low energy end of the spectrum. And those photons, when they're absorbed by a molecule, they cause things to move around. And so one of the ways to excite an electron to move from one energy band to another is by putting in heat. And this reminds me of one other thing one other speculation which I want to say, since you're all mostly asleep and won't hear this. What is the meaning of higher levels of consciousness? And it turns out, and this is basic physics, that there are many energy bands to a molecule, lots of them. Let's shrink this down. Here are a bunch of energy bands that are occupied by electrons. Okay? There exist higher energy bands, I'll draw them with dashed lines, which are unoccupied. Up here. And up and so on. These different energy bands they exist because there are many different orbitals, they're referred to as orbitals, different ways that an electron can exist inside of a molecule. And I suggested to you that it may be the movement of energetic electrons from the highest occupied band to the lowest empty band, and the reversion back down again that produces light, light photons that could be part of the aura. Well, it's conceivable that electrons could also be excited in some way to higher orbitals, to higher energy levels. And since there are lots of these energy levels that are available, these then could correspond to different, that is, the flow of energy through different energy bands, higher and higher energy bands, could produce a very different, consciousness, if you like. So there's a speculation for you. Yeah.
What effect does, I don't know if you know, but what effect does gravity have on the flow of electrons along these energy bands?
Okay, what effect is, I don't know the answer to that. What is the effect of gravity on this? I have not, maybe Gio has the answer to that.
Jim and I were kicking this around yesterday, and I think what both of us is probably the mechanism for that. Constantly, as we're shifting around in our whole body with gravity, we're changing the mechanical stress in the soft tissue.
Gio, could you mention this on the microphone or somehow so I can get on the tape? If
you're going to...
Why don't you come up?
Okay. I want to keep this very brief because it's another whole lecture. This is what it says. Basically, and what was so exciting about Ron Kirkby's model of taking synergetic principles and seeing those in human spine is that he was beginning to look at a way of getting a geometric form through which those stresses are distributed. Now, what is known about collagen is that when you tug on it, it will start producing electrical fields, and what Jim has given you is a very detailed explanation or mechanism through which the fields that are found around large sections of collagen when they're stressed are probably generated. So he's been working down in the molecular level, and I'm trying to talk about it at a slightly larger level. It is of a piece of collagen maybe 3 inches long and a half an inch wide or something like that, which are the kind of experimental specimens that they play with from bones. And they also have done this with tendon. And as Jim said, you can talk about the level, the molecular level or intramolecular level that he's talking about, and talk about the macro level at the same time because of this incredibly orderliness of these quarter staggered arrays. So whether you talk at that level or you talk at a larger level, you can use the same principles. And interestingly enough, the empirical evidence all seems to jive too. When you put stress on collagen, particularly shear stress against the longitudinal axis of the collagen fibers, you can read out what is called a Hall effect voltage. But which is evidence that there is a flow of electrons in the longitudinal axis of the collagen. And I have a hunch that where gravity comes in to all of these other processes is it's constantly changing these mechanical stresses, which is shifting through the mechanism Jim has outlined, these conduction bands. Now it's interesting because Jim has picked up on Valerie's work, and particularly the notion of auras and light, and our orientation is more funky, and we want to think about gravity, and really I think you have a whole bunch of different stresses coming into the system, or inputs coming into the system, all of which can get electrons moving. Another one is pyroelectric, Gladys, because it's been shown that the collagen samples, when they're heated in certain ways, will again evidence this Hall effect voltage. There will be a flow of electrons that can be measured. And photoelectric as well. So whether it's heat or mechanical pressure or light as an input, you can read out electricity from collagen. Now what's been very interesting to me as I've thought about other forms of healing, particularly laying on of hands, is that people describe, as did Amos when he talked about sending electricity through a body when he does some work, and then the tissue is much easier to work on. Now, that may all sound very subjective, but we've all had an experience, I think, or a number of us have had that experience. And I have a hunch that when people work with laying on of hands, they may in fact be generating an electrical field, a net voltage differential between their hands that's out of the ordinary, that they can sustain for a certain period of time. And that will change the mechanical shape of the collagen directly. Now, it's going to be a while before we can nail that down in a lab study, but I think there are ways we can do that on the macro level of the whole body, and I'm hoping still that the work that Dr. Burr did and that measurement of the electrical potential at the surface of the body will be a way we can get a handle on that. If we could instrument that input or that output from a body while it's being rolfed and look at the mechanical pressure that a rolfer is applying through their fingertips, and instrument it in such a way that we can see what we're putting in terms of mechanical pressure and what we're reading out in terms of a change in electrical field throughout the whole system, we may have the elements in a laboratory study to really answer your question and get a handle of what we're doing in our rolfing. Thank you.
I don't know what gravity is. That's my problem. And it seems to be the physicists don't either. And that's, here we are. I mean, it is a pretty important force in our existence. And Just goes to show you how little we actually know. Yeah.
This question, and it may be silly, but visually what I see in this spiral is a tube effect. So ultimately it's a tube with an opening. Okay, I see visually that the three strands spiraling create within them a hollow center. And when that is stretched, perhaps there is something in that hollow center, if it's just amorphous tissue fluid or whatever, but when it is stretched, whatever is in there, whether it's nothing or something, is going to get squeezed out, have to go someplace, because the space is reduced. Does that have any?
Presumably, there isn't anything in there.
Jim, it was once offered by one of the other Rolfers in the Institute, not myself, that there was a possibility of cerebral spinal fluid passing through those hollow channels. Is that feasible in your mind?
You're talking about a hollow channel on a much larger level. This is just a molecular level.
Yeah. It's space that can't be occupied because the reason that the molecules are apart is the bond structure already. I mean, it's the space is already used up by the potential energy field. There's no place to put anything inside.
So that was in relationship to fibroblasts then?
I don't know, whatever.
Yeah.
Basically, the question about what is in the space between molecules is the question that physicists have oscillated up and down on. And they used to call it the ether. And then they did an experiment which showed there was no such thing as an ether. So I don't know what is in that space, but I can tell you what there isn't in that space. I can tell you, for example, that there isn't any water in there. And water is the smallest molecule. Well, now there could be dissolved gas. There could be gas in there. There could be oxygen, for example. Hydrogen. Well, oxygen,
Yeah, I think it's important to comment that although the official paradigm in science says that the ether, or notions of an ether, have been beaten dead a long time ago, some of the most important scientists at the turn of the century, including one of the two who designed that crucial experiment that supposedly killed the ether, worked for another 30 years of his life on revised theories of the ether. And it's another one of those underground or lesser-known theoretical developments, and I'm not convinced at this point, from some of the reading I've done, that an ether proposition about that intermolecular space is completely ruled out. There's one place you can get a kind of layman's access to this. I don't know what to make of it, and I haven't found any physicist friends who are willing to comment on it. But Patrick Flanagan, who's the young man that's done a lot of work with pyramids, has something he calls an ether vortex theory, which he talks about in the end of his little book, in which he discusses some of the anomalies that were left over from this Michelson-Morley experiment that supposedly put the lie to ether, and puts out his point of view about it.
Yeah.
Could you just say a few words on the relationship of energy being transmitted to connective tissue, collagen fibers, as opposed to being transmitted by acupuncture meridians or neural pathways?
Okay, the question concerns the relationship between the electron, the flow of energetic electrons, and the acupuncture meridians. And it's my understanding that the acupuncture meridians do not follow the nervous system. Is that right? They're totally... So, the only thing I could say is, since acupuncture does appear to affect the flow of energy through the organism, that these meridians that they could be differentiations of the tissue, specializations where this energy flow is channels for the flow of excited electrons. So that's a possibility. Does that answer your question?
And we normally speak of energy flowing normally, I mean, in unsophisticated terms, through neural pathways. How muscles are excited or activated. Described in the text as occurring through neural pathways. So that kind of blows that out.
It's a different thing.
The question concerns the function of the nervous system, I suppose, and according to conventional physiology, which this seems to be pretty well worked out, the action potential travels down the nerve and reaches the muscle and stimulates the muscle to contract. Now, the question is, where does the energy come from for that muscle to contract? And this is another one of the unsolved problems in biology. Where does the energy come from muscle contraction? And physiologists don't believe that the energy comes down the nervous system, through the nervous system. That supposedly is the signal. The energy should be manufactured locally within the muscle by metabolic processes, according to conventional biochemistry. But there's something real wrong with that, because ATP is supposed to provide it's supposed to be the magic energy molecule of life, and is supposed to provide the energy for the contraction of a muscle. But muscle biochemists have had the serious, embarrassing difficulty that they can get a muscle to contract furiously, and there's no change in the ATP level in the muscle. So the energetics of muscle contraction. In fact, this is one of the reasons St. George switched from working on muscle to working on electrons, because of this dilemma with the energetics of muscle. And I think he now thinks that the energy flow is via these excited electrons.
Specifically collagen.
Well, collagen is one of the proteins that St. George has studied. And he has found that it is a conductor of electrons. This seems to apply to other proteins as well, not just to collagen. Yeah.
Spinning metal needle affect electromagnetic field?
Would a spinning metal needle have an effect? If it were magnetic, sure. Even if it weren't magnetized, it would have an effect. Yeah. All materials affect magnetic fields. Some of them enhance the field, and some of them decrease it.
Then those fields in turn would affect the electron.
The field would in turn affect the electron flow? Sure. Okay, Lloyd?
Has he seen the report? Has St. George seen Dalton's report?
Gee, I don't think so. Would you like me to show it to you? I feel that the difficulty with the report is that the aura is not presently within the province of modern physiology. So the report in its present form, if shown to a physiologist or a medical person, would not, they would say, they would look at the art and it's not in Guyton's medical physiology book. So
that's true, but also some of the things you're saying lead to the possibility that you might pick up auras that are very sensitive to a number or something that maybe no one's tried. You might turn him on to looking for auras.
I might.
The model that says that there may be a physical measurement possible.
Yeah, I think the overall thing of why I'm here, I think, is the relationship between Rolfing and science. And everywhere I've looked so far, every phenomenon that Rolfers have told me about and that has been referred to in the Hunt report seems to fit with what the physicists and the physiologists have to say. I mean, I don't, and this is as it should be. I mean, it should be harmonious. I mean, there should be harmony between all of this information that we have from all this work that's been done for hundreds of years, or thousands of years really, since scientists first started to consider what life is. And the work that you people are doing, which seems to have such vast effects on the total organism. So my approach is to look at the literature very carefully and try to find out, to locate these places where things agree. Yeah.
Something that I thought of a moment ago that intrigued me was the relationship of how the traditional view is that energy or communication flows through the channels of the nervous system. And I'm seeing how this model you're discussing is bringing up a whole other model of how energy flows through the connective tissue network.
Yeah, I think it is. I think this is a...
Could you comment on like the relationship of those 2 points of view? It's looking at that.
The antenna.
The antenna.
Back to the body being an antenna.
Yeah, it does. I mean, the collagen molecule really does look like an antenna to me. One of the other unsolved problems in biology is communication between organisms. And this is off the subject of your question, but I'll say it anyway. I have a colleague, Frank Brown, who is a very distinguished professor at Northwestern University, who has been studying the communication between organisms. And he's been studying seeds, plants. And he's found, and this has profound implications for ecology, that 2 seeds allowed to fall, basically this is what he says, 2 seeds that fall down next to each other, hit the ground right next to each other. One will grow and the other won't. Well, this doesn't always happen, but statistically this is what happens. And he has studied this phenomenon very carefully for many years, and he has discovered that seeds communicate with each other by some mechanism, and he has found that this communication, whatever the force, whatever the energy that flows from one seed to the other, can get through what's called a mu metal shield, a shield that will block electromagnetic radiation, but not magnetic fields. So he feels that all organisms communicate with each other via what he calls biomagnetic fields. And he has very good evidence for this, and he's published his findings in several different journals, which I can refer you to. So this, and he feels that he has demonstrated that a population of organisms will all be in communication with one another. And it seems like very sound data.
One last bubble on that general territory is that if in the animal territory the collagen is the network of, or at least we're looking at collagen as a network of electronic communication or energy fields of the territory. In the plant territory, might cellulose serve the same function? Has anyone looked at cellulose?
Sure. Well, there are a number of similarities between cellulose as the extracellular matrix or extracellular structure forming element of plants and collagen. But I don't want you to get too focused on the collagen in the sense that I feel that we have to remember the other structures of the organism, which are also highly ordered. For example, the musculature. The muscles produce an X-ray diffraction pattern. The proteins of muscle are like collagen, very long, thin, filamentous proteins, and they're organized in a very precise way, and the organization changes during contraction. So muscle is likely to have similarities in terms of its its field properties and its conduction properties. It's the possibility that some of the aura, some components of the aura, arise from the musculature, from the bone, from the circulatory system, from the nervous system itself. Yes, sir.
Could you tell us what you're going to talk to us about tomorrow? And I'd like to wrap it up.
Okay. Tomorrow I'd like to talk about the rest of the connective tissue, the mucopolysaccharides and the material between the collagen molecules. And I think I have less to say about that. So.
Well, I'd like to say that I think there's no one here who doesn't feel happier because of what you've said today, because this problem of the connection between Rolfing and science has been a matter of concern to all of us for many years. So we welcome you doubly for your validation of our work. Thank you.
Party and dance at 8 p.m. and by that time I'll have the locations of the multiple sessions. Sometime this evening I'll have the locations of the multiple sessions posted for you.
Will the multiple sessions all be taped?
I doubt it. Thank you.
Basically, like Bob New York can produce himself in Tripoli, they'll also
The party is right here.
Greater Rolfing Boogaloo Review Band. That's set up this
Hi, Charlie. Great. I got it. So, I'll be here. Good. Today,